Hello everyone, my name is Meena Ramchandani. I'm an infectious disease physician at the University of Washington in Seattle. This podcast is dedicated to an STD literature review for healthcare professionals who are interested in remaining up to date on the diagnosis, management, and prevention of STDs. Hi everyone, welcome. We are lucky to have Dr. Daniel Rocky and Dr. Scott Grieshaber join us for this episode. Dr. Rocky is a professor in bacteriology in the Department of Biomedical Sciences and the Carlson College of Veterinary Medicine at Oregon State University. Dr. Scott Grieshaber is a professor in the Department of Biological Sciences at the University of Idaho. Both of their research focuses on chlamydia pathogenesis and the interactions between chlamydia and the mammalian host. And this episode will focus on an article titled Metabolic Dormancy in Chlamydia trachomatis treated with different antibiotics, which was published in Infection and Immunity in February of 2024. Welcome, both of you. We're so excited to have you on the show.
Thanks so much for the invite.
Yes, good morning. Good to talk to you.
Now, this will be more of a basic science session. And to orient our audience, can you talk about some of the aspects of the microbiology of chlamydia and how it lives or replicates in the human host? And let's focus on Chlamydia trachomatis for this episode, although I understand we might digress to other chlamydia as well.
Yeah, chlamydia is an obligate intracellular bacteria. That is, it has to live inside a eukaryotic host cell. And this is true of a long evolutionary line of bacteria in the chlamydia family. Goes back roughly, I think, over 1 billion years. There was a good paper that described that a few years ago from Matthias Horn's lab. But essentially, they grow as parasites. They have to enter a eukaryotic cell, and they set up a replication niche where they go through a developmental cycle where they change phenotypic cell forms. They enter as this small cell form that's infectious. It's called the elementary body. It's very small. It has this cross-linked outer membrane, and it's highly condensed nucleoid in its DNA structure. And that makes it a very, very small cell form. This cell form enters the cell and starts the process of infection. It needs to go through this germination process where it becomes this replicating cell, the RB. We call it the reticulate body. The reticulate body expands the infection, so you get more organisms. And then those expansions of those organisms then produce more EB cells to then disseminate the infection. So chlamydia separates expansion from dissemination. And after chlamydia replicates to high numbers of creating these infectious forms, they then can lyse the cell or egress from the cell. It's not quite clear exactly what percentage of the time they do each. It's something that's hotly debated. But in the end, these EB forms exit the cell and then go find new hosts, new eukaryotic cells to host. So basically, chlamydia goes through this cycle where they find new hosts, enter, expand, and then go find new hosts again. And to do this, they go through this, like I said, this cell form developmental cycle, producing this replicating form, the RB, and this infection form, the EB. I'm sure I left something out, but I'll let Dan jump in.
Thank you, Scott. Yeah, Dan, do you have anything to add?
Well, I think it's interesting to think about the idea that there are chlamydia in almost every animal out there, and they're different. As Scott said, the lineage goes back for at least many millions of years. And remarkably, when individuals went out in the Alvin submarine and went down into the depths of a trench near Iceland and took a scoop of muck from the bottom of that trench and just did metagenomic sequencing on that, the number of chlamydia-like organisms in that was really quite surprising and filled up every lineage of what we call chlamydia nowadays. So it's pretty remarkable how long the lineage has been around and how successful it has been.
Exactly. And they all share this developmental cycle.
Oh, wow. That's fascinating. So goes back millions of years, potentially, or hundreds of thousands of years, and all share this kind of same pathogenesis where they have this cycle of these different bodies that it resides in the human host, the elementary body and the reticulate body, correct?
Correct. I think that certainly in the sediments, it's not clear exactly how the life forms are perpetuated. But in every contemporary chlamydial species, the organism exists intracellularly in a vacuole. It has a developmental cycle. In some chlamydia that occupy niches inside protozoans, I think they might be as more commensal as opposed to strict pathogens. I think that sometimes the host will grow better with the chlamydia inside.
Yeah, like Dan said, not every host has been defined. So some unknown, whether they have a host or what the host is.
Now, how does that relate to chlamydia persistence in terms of like the reticulate body, the elementary body? Can you describe chlamydia persistence, and does chlamydia trachomatis demonstrate persistence?
When I think of persistence, I always want to think in terms of clinical and basic biological terms. Certainly, in virtually every animal that has a chlamydia infection, persistence is a major part of that natural history. In sheep, for example, chlamydia abortus persists through the life of the animal. Once it acquires it, often as a very young animal, but only manifests disease when a first pregnant ewe is in the late stage of the pregnancy. Pathogen emerges from persistence, then grows aggressively in the placenta, leading to death or morbidity of both the ewe and the fetus. In humans, persistence certainly is a part of the life history, and many individuals who are infected asymptomatically, and the pathogen exists as almost a commensal in the host, that the pathogen exists without causing disease in the host. And in the laboratory, chlamydia can be induced into persistence via treatment with any number of different stressors that affect very different aspects of growth. And one of the defining aspects of this persistence is the generation of viable but not culturable, unusual developmental form that are called aberrant forms. As Scott mentioned, elementary bodies, the infectious form, are very tiny. You can fit hundreds of them inside a vacuole, inside of a cell. Now, aberrant forms tend to be much larger than that. They often approach half the size of the nucleus. And these are organisms, live chlamydia, that will make DNA, make protein, establish their niche, but they never, in the presence of the stress, they don't mature to an active infectious elementary body. Take the stress away, and through mechanisms that are really unclear, this persistently infected cell will then generate new developmental forms, new elementary bodies or reticulate bodies that can process through the rest of the developmental cycle. So chlamydia can be seen as persistent pathogens in patients, and we can demonstrate persistence in laboratory cultures.
That's perfect. Thank you. It's almost stress. The organism goes into this aberrant form to kind of tolerate that stress. And once that stress is removed, then it can go back into the replicative kind of life cycle. Is that correct?
I would just like to stress that what Dan said is really important, that persistence in infections is absolutely clear, right? We do know they form these persistent infections, and we're trying to model that in vitro as best we can. And so the best model we have is this aberrancy effect. But we really don't know a lot about how this works and how this works in relation to this persistence during infection. So if you look at other bacteria, right, persister cells are these forms that are usually low metabolism, not replicating. And so therefore they're not great targets for antibiotics. They survive an antibiotic onslaught because they're not doing anything. And so chlamydial persistence has always been on the other end of that. And this is aberrancy, this idea that there's these cell forms that are staying sort of stuck in a part of the cell cycle that's active, right? They're actively making proteins, they're synthesizing DNA, but they're not actually forming the infectious progeny. And so that's our current model, and it's called aberrancy. And it's the best model we have. But I think there are other aspects of chlamydial biology that lead to persistence. This idea that some forms of chlamydia are true persister cells. Like the EB has really low metabolic activity. It makes energy, so it makes ATP, but it doesn't really transcribe or make a lot of proteins, and it doesn't divide. So it's already resistant to antibiotics that target cell division. So there's kind of this dual aspect of chlamydial persistence, that we know we have this clinical persistence, and we have these models in culture that we're trying to sort of understand how those relate to clinical persistence. And one of these is aberrancy. And the other side of that is the fact that the chlamydial EB itself has these persister cell-like phenotypes.
What kind of stresses instigate the aberrant form or chlamydia persistence?
So it's kind of surprising how many ways you can make chlamydia go aberrant. And certainly we talk about antibiotics, but certainly this is a means for survival inside cells that has evolved over the course of certainly the pre-antibiotic era. Okay. Stressors such as low amino acid availability, immune mediators such as interferon gamma, and starving cells for molecules such as iron can all drive chlamydia into persistence. In fact, I think if you can stress the chlamydia in any way that's non-lethal, they will tend to go into this default aberrancy pathway. And it's not clear exactly what's going on here, because what you have is this microbe that was so small to start with now is really quite large and occupies quite a bit of space, a single microbe within the cell. And this is documented in vivo in patients' tissues from hysterectomy patients. They can see aberrant forms in vivo. But the function of it is not super clear. Why does it need to be so large? That's the great question, I think, in aberrancy. And what we'll talk about, Scott has generated a lot of very nice video microscopy images that don't necessarily add clarity to that, and then try to say what is really going on in this aberrant form inside cells and what is its role during the infectious process. One very interesting inducer of aberrancy is co-infection with herpes viruses, in which a single herpes virus protein has been shown to facilitate the development of aberrancy inside cells. So the interaction between the herpes and chlamydia, which probably goes back a long, long ways as well, will lead to aberrancy within infected cells. And now I don't know what happens in vivo, but there's solid in vitro data on that one.
Has there been any other association with other organisms like that?
So people have tried to co-infect cells with chlamydia and a lot of other things. And remarkably, to me at least, they always seem to ignore each other. If you put Coxiella burnetii, which is another obligate intracellular bacterium, into a cell and chlamydia in the same cell, they will form individual vacuoles that never seem to talk with each other. Toxoplasma gondii has been infected with chlamydia. The toxovacuole is morphologically perhaps as similar to the chlamydia vacuole as anything, and they don't interact at all. And that's a eukaryote with a prokaryote. Even within the genus Chlamydia, if you put Chlamydia pneumonia into a cell with Chlamydia trachomatis, they appear to not interact directly at all. They form individual vacuoles and have their own time scale. So they seem to ignore each other inside cells.
So, Scott, I want to ask you a question. A term has come up, metabolic dormancy. And so what is metabolic dormancy in the setting of Chlamydia trachomatis or other bacteria? And is this the same thing as latency or persistence? How do these terms kind of interact, or are they different or the same?
So latency would be like the classical, herpes, even HIV, right? That basically the virus hides out completely and makes maybe a few transcripts, but very little effect on the host. And, in the bacterial world, we don't really think much about latency. We think more about, like you said, persistence or this metabolic dormancy, the fact that the cells themselves maintain their structure, they maintain at least some level of protein synthesis to maintain their energy levels, but at the same time don't cause disease. So they're kind of ignored by the host because they're not actively destroying tissue. They're not doing this. So it's sort of equivalent to the latency in the viral world. For chlamydia, this is like just part of the issue, right? We know we have this persistence clinically, and we know we have these aberrant cells, but then we also have, like I said, the EB form, which is, in my mind, less like a spore and more like the dissemination form. So it's a cell form that's only job is to go find a new host. So I don't know how much you know about microbiology, but there's an organism called Colobacter crescentis, which is sort of the model system for swarmer cells. So cells that undergo a developmental form and then go find a new place to live. And I would say the EB is this metabolically active cell form that's maintaining its ability to infect cells, but it's waiting to find a new host cell. It doesn't divide, but it needs energy. So we showed a few years ago that it needs ATP or glucose 6-phosphate or amino acids, something it can use for its the TCA cycle, so it can generate its own ATP because it needs that energy to infect cells. So chlamydia isn't quite a spore, but it's more of this disseminator cell that can keep itself alive for some time scale. I think we measured it on a time of 48 hours with an energy source that can find a new host to infect. So the role of aberrant cells versus these EBs versus this other form, which we sort of maybe talk about this paper, chlamydia who have entered cells but then not fully germinated, is this other form of low energy cell that doesn't seem to be doing much, but maintaining its energy pools, maintaining its ability to eventually grow out again.
That's helpful. And do we find, or is there any evidence that all the chlamydia change into the aberrant form? Or, for example, can they exist some in the aberrant form and some in an infectious form?
The developmental cycle is complex. When the EB touches a host cell, it enters through this type 3 secretion system mediated endocytosis. So it drives its own endocytosis by directly reprogramming the cell, enters the cell. And in the model system we use, which is chlamydia L2, this takes 10 minutes to get inside. After that, they go through this germination program. It takes about 10 hours before they start replicating. And then after replication, they start producing these intermediate forms, which then transition to EBs. So as soon as 18 hours inside the cell, you have a mixed infection. You have both RB cell forms, you have IB cell forms that are going to become EBs, so those intermediate forms, and you have a few EBs. So depending on when you treat them or when they get the stress, those inclusions will have all the cell forms. So you will have aberrant cells, but you will also have EBs in these cells, those infectious forms. You have these mixed environments and then last hours. I'm not sure we actually tested how long the aberrant form and the EB can coexist in the cell. We did some experiments where EB that forms, say, at 18 hours post-infection can still be there 30 hours later and still infectious. So that EB still collects inside this inclusion. And if you give them a stressor, those EBs aren't affected by the stressor because they're low energy metabolism. I hate the word dormancy, but they're these persister-like forms, right? They maintain their energy levels, but that's it. They don't replicate. They don't synthesize a lot of protein. They're just maintaining themselves for release. They're basically just waiting until they can get out. So if you treat those cells with a stressor, you get aberrant out of the RBs, but you also still have the EB forms present. So you always have this mixed environment as soon as, say, 16 hours post-infection. So to answer your question, yes, you will get aberrants, but you'll also have EBs present most of the time.
I will say that in vivo, in patients, there's probably all forms present at the same time, certainly in the patient and probably within some single cells. If you grow chlamydia in optimal conditions in a laboratory and you look at a bunch of cells, you will find aberrant forms. They're present in normal infections as well. And that's probably because there's some little twitch of stress in those cells and it's driven them to aberrancy. So yes, you can find each of the developmental forms, certainly in every patient probably, and I would wager almost in many different individual cells.
Thank you. This was a really helpful overview to give a background for our audience. Thank you so much, Scott and Dan, for this really informative episode. This has been such a great session, and I learned a lot from speaking with you both.
Thanks so much for the invite.
Thank you very much. Good to talk to you.
This podcast is brought to you by the National STD Curriculum, the University of Washington STD Prevention Training Center, and is funded by the Centers for Disease Control and Prevention. Transcripts and references for this podcast series can be found on our website, the National STD Curriculum, at www.std.uw.edu. Thank you, everyone, and have a wonderful day.